Hi, everyone. My name is Frances Grace Stark. I am a PhD candidate in microbiology at the Krasileva lab at UC Berkeley here in California. And today I'll be talking to you about my thesis work in elucidating the fungal immune system. Okay, so a little bit of introduction. I grew up in Austin, Texas, and I started a bachelor's of science in biochemistry from UT Austin. And so, Central Texas Mycological Society is kind of my home base, even though it started about the time I left Texas. And so it's just been great to see it grow. Anyway, so while I was doing my undergraduate, I participated in undergraduate research, which has always focused on how organisms interact with one another. I first started with fungal endophytes in leaves of Texas native grasses. So my kind of first research topic was on fungal ecology. And that just opened up an entire new world. I then started focusing on viruses and plants. And so I've always been interested in plants as well, as you can see of me highlighting plants over here. And I then, after college, started to become super interested in fungi through foraging and naturalizing, specifically in Texas. I've done a lot of naturalizing in Texas. And so, oops, sorry. Ooh, go back. Not that far back. Okay. And so I graduated from UT Austin and I moved to California for my PhD in microbiology. And I'm currently extremely interested in cell and molecular biology of fungi. And so this is kind of showing what I've gotten up to in California since moving, about two and a half years now. These are very specific California plants. And my first study in graduate school was post-fire fungi. And so here's me burning a forest up here in California. There was a bunch of, field sites that we burned, and we did ecological studies on what would show up after a fire. And so I just published on this work. And these are very specific fungi that grow in the subnevian zone in the Sierras. And so I've had the honor of seeing them. And so, just a bunch of naturalizing still is kind of the hobby. And here's me foraging for chanterelles. So yeah, and that's where I'm at. Okay, so first I wanted to give a little bit of an introduction to fungal biology. So many fungi spend most of their life as vegetative mycelium or yeast. I know that I'm glad to be speaking to a mycological society, so I don't have to go too in depth with this. Y'all kind of know. And they only produce sporocarp, so mushrooms is an example of a sporocarp after sex. And so the cell type that makes up most filamentous fungi is the hypha. And fungi are extracellular heterotrophs, meaning they cannot make their own food, like plants, basically. They need to scavenge their own food. And they do this by excreting enzymes out into their environment extracellularly and then uptaking nutrients that they break down. Filamentous fungi arrange their hypha in linear circular arrays. And so they have this kind of circular growth pattern. And they extend at their tips and are able to branch and fuse, leading to a loose three-dimensional network called mycelium. And this enables fungi really to maximize surface area for nutrient absorption and enables fungi to effectively colonize and reach new substrates, having this elongated cell form. A sporocarp, which means the tissues that produce a spore, is usually just compact and differentiated mycelia. So as you can see, this picture kind of shows that the cell type of fungi is really this hyphal mycelia form. Even in higher order structures like the mushroom, you can see that it's just arranged mycelia in different ways. And this produces the spores. So these are the gills with the basidia specifically for the mushroom forming species. And these spores are able to effectively colonize new sites. And so, yeah. So what is cell and molecular biology? It's kind of the field of study that I'm in. And so I kind of wanted to go over exactly what this means. And so cell and molecular biology studies the structure and function of the cell, which is the basic unit of life. So here I'm showing a fungal cell. And many questions in basic biology, for example, how does a fungal mycelium grow, period, can be answered by observing and manipulating cellular processes. And basically this, you can think of this kind of as answering two questions. What components make up living cells, and how do the cells function given these components? And so here are specific examples of more specific questions. And the more we figure out, the more detailed questions we can ask about the machinery of the cell. So how does a cell develop and grow? You know, how does a cell recognize and uptake nutrients? How do cells change these nutrients into energy that, you know, is used to help grow? How do cells interact with one another? And, you know, how do cells function in times of stress? And many of these questions are not mutually exclusive. So usually if there's some sort of pathogen attacking a cell, the cell changes the way it develops and grows. If the cell is, you know, has different nutrient sources, it changes the way it interacts with this environment. And so it's a pretty big field of study. And so how do scientists dissect the workings, the inner workings of a cell? And so ultimately genetics, which means to disrupt the gene. So I'll take a step back because I'm not sure our familiarity with this kind of stuff. So a little Bio 1A refresher. Most cell components and molecules are created by proteins, which come from the genetic code, DNA. And so disrupting this gene leads to loss of a function of this protein. And proteins are typically used to make up most of the cell, even if it's not directly related to the cellular function. Usually a protein is used to help create this function. It just said participants can now see your screen. Were you not able to see my screen before that? Or are we good?
We've been able to see it the whole time. Okay,
cool. So this is classic ways to figure out what's going on in the cell. So here's an example. So I also just wanted, anyway, I forgot to kind of explain what was going on here. Basically, I just wanted to show how dynamic a cell is and how crazy the biology is. This specifically for fungal cells, this specific apex of the hypha is called the Spitzenkorper, and it helps to deliver new growing machinery to the fungal cell. And so basically I'm using this as an example here. So we have confocal microscopy, where basically these little green dots are these specific proteins that we hypothesize to be useful in the growth of the cell. And we have attached a green fluorescent protein onto our proteins of interest. And then we do microscopy where we see that these proteins localized to the apex of the cell. And so now we can hypothesize that these proteins are important for fungal cell growth since they're in the Spitzenkorper. And so basically what we do now is genetics. We knock out each one of these genes and see how the difference of growth, we see the difference of growth. And so basically this is showing that wild type here with no genetic knockouts grows fine. And relative to the wild type, you can see this little delta right here is the knockout. And so we knocked out these genes and we saw a reduction of growth. And so now we can kind of hypothesize that these genes are necessary or needed for growth of the fungal cell. And so this one looks to be like it was necessary completely. Like if you knock out this one, you lose all growth. And so fungi, like all other cells, are constantly interacting with their environment. So cells are quite literally the products of their environment. And so they perceive their environment and usually by highly complex signaling mechanisms that are specific to how the cell or where the cell finds itself. And cells usually have a very specific responses to very specific environments. And this can change the entire biology of the cell. And so it's really a complex, like biology is truly infinite and like how things function. And so the ways that people figured this out is you can knock out with the genetics that we just discussed, each section of this response, and you can kind of dissect exactly how the cell is interacting with the environment. Sorry, let me change this here. And so here I'm showing you all the different environment factors and how they change the expression of this one gene called ochratoxin A, this OTA gene. And it's a type of mycotoxin that is produced mainly by Aspergillus and penicillin species, and it's classified as a carcinogen by the International Agency for Research on Cancer. So it's a big thing that people study because, you know, Aspergillus and penicillin is everywhere, and it's a big carcinogen. And so this just is exemplifying all of the signals that a cell uses and how intricate the response can be to all of these different signals and how it can affect like very, very certain gene products and how things are formed and regulated. And so now that I've gone over the basics of cell and molecular biology, I kind of wanted to open up a question to the floor and see what y'all had. You can probably just write it in the chat if you wanted to. But why might you want to study the cell and molecular biology of fungi? Like, why would it be important? You can use this example if you wanted to, of this carcinogen being created, but why might it be? There's a lot of things going on in the chat. Ooh. Oh, cool, someone's up in the bay. Why is it important to understand what's going on in a cell? I don't know. Basically, basic biology is the main reason, like understanding how, to understand how environmental conditions affect everyday life. Okay, I'm getting a lot. So we can understand that technology and apply it elsewhere. Very true. Okay, so I'll kind of go over these. Environmental conditions, like basic biology, just understanding how cells function in different environments. How we can understand that technology and apply it elsewhere. And this is really important in biotechnology with fungi. If you understand the machinery that fungi have within themselves, you can understand what products they might be able to create. You can then put these proteins in these fungi. Oh, sorry, let me go back, please. You can infer what's going on in the organism. Yes, that is the main thing, is just basic biology, understanding how a cell works, 100%. We want to understand fungal cell biology. Yep, basic processes of mushroom life, exactly. So like, when does a mushroom even form and how does it form? Use the understanding to manipulate or optimize organisms. Exactly. A lot of people do this with plants. They want to optimize photosynthesis. And so understanding exactly how photosynthesis works, you can kind of like start to, you know, manipulate that. Yep, their answer. So there are still questions we don't have answers for in regards to fungi. I'll kind of go over what we don't know. And the answer lies in the building blocks, 100%. So exactly how the cell is functioning, how the cell functions with itself. All of the above. Figure out how to put them to work for us. Yep, this is with the biotechnology, kind of understanding how to manipulate fungi genetically and how, you know, so a lot of people are trying to put hemoglobin in fungi for, you know, sustainable food, kind of like how they did in impossible meat. And fungi produce their own hemoglobin. And so you can just upregulate their endogenous hemoglobin, and it's pretty cool. And so I'll kind of end it here. When mushrooms appear, very, very true. A lot of people like can see if there's certain levels of plant hormones that are near like ectomycorrhizal fungi, they'll like have more fruiting and blah, 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 a bunch of ecology work. And so, this is good. Yeah, good inferring. This is exactly everything that I think of when I think of why it's important to study cell biology. It's just like, it can help you really infer why the organisms are where they are. And it just gets to the basic questions So yeah, I mean, cells are pretty convoluted in how they function. So it's sometimes harder questions to answer, but it's definitely why you would study it. Okay, cool. And so, okay, next, I'll close the chat. Okay, so like we all just kind of talked about, in the field of fungal biology, there is much, much more to learn. And so in the first Basidiomycete genome was published in 2004, which is just crazy because now we have about 1,500 genomes sequenced in fungi. And then, you know, that's still like a pretty small number compared to like plant genomes and vertebrate genomes. And so before this, yeast was published because yeast has been used, you know, in biotechnology for forever and basic biology for forever. But yeah, basically, I mean, it wasn't even until 1969 that fungi got their own kingdom, even though they were studied pretty separately. You know, this is the era of fungal biology is kind of now, especially with like new sequencing technologies. So I just kind of wanted to talk about some of the numbers. So we have roughly only described about 1,400 species. So this is of 2020. And it's estimated, or this estimate was in 2020. And it's estimated that there are 2.2 to 3.8 million species. And this estimate was made in 2017. And we've only sequenced about 1,500 genomes, like I just said. So it's you know, there's still so much to discover. And specifically when it comes to genes, when we look at these fungal genomes that we now have sequenced, we realize how little we know about these genes. And so the estimate is about, there's about 70% of the genes on average in fungal genomes have no homology to anything else. And so this means that, you know, we have mitochondrial genes or ribosomal genes that we can see in fungi because it's a highly conserved process. And so you can kind of infer that the genes, even though they haven't ever been looked at like under, you know, any cellular and molecular biology studies, you can infer that these genes are doing the same thing. However, most of the genes in fungi, you can infer nothing, like it matches to nothing in the tree of life. And so there is so much to discover. So I urge you to take up a career in fungal biology. Okay, anyway, so a little bit, now that we've talked generally about fungi and cell and molecular biology, I'll kind of go into what my lab does. And so my lab specializes, here at UC Berkeley, specializes in studying the evolution of innate immune systems. So I wanted to kind of go over that. So immune systems are broadly defined as biological processes that protect an organism from invaders. And that's it. That's basically the main definition. And all organisms interact with other organisms perpetually. Like we have bacteria living within us. And so immune systems are vital for the survival of organisms. And so here I'll go kind of over the classification of immune systems as we think of them now. And so immune systems can be classified as antagonistic-independent, which means that protective processes are on all the time, or antagonistic-dependent, meaning certain protective processes are only turned on when an invader is present. And so basically, antagonistic independent it's more thought of as protecting an organism for a large range of microorganisms. And so you always have organisms that want to enter your cells, but because we have skin cells that are tightly compact, a lot of microorganisms can't infect us. And this is the same with bark on a tree. That's why bark exists. And there's other products like amatoxins and psilocybin that are produced all the time. So these genes are always on, they're always producing, and it's supposed to, you know, knock out anything that tries to eat it, right? And so antagonistic dependent is more of a recognizing pathogens more specifically on a more like cellular level. And so I'll kind of go over this a little later, but basically we have recognition of a microbe that's trying to enter the cell, and we have an upregulation of genes to kind of combat this microorganism. And so this inducible nature really allows the cell to conserve energy by only turning on responses when necessary. Specifically, if there's a specific microorganism that's particularly deadly, like a bad virus or something, this can help be another layer of protection. Okay, so antagonistic-dependent immunity exists in all organisms. And this can be separated again into two categories. So now we're only looking at the cellular and molecular biology, not the like tissue organization, right? And so these categories are innate immunity, which is most organisms, and adaptive immunity, which is vertebrates only. And so most organisms, including us, have innate immunity, which means it's usually an immediate and general response to pathogens, and it's found in all of our cells and tissues usually. So every single cell has these very certain products that are there to recognize pathogens, and it does not increase with repeated exposure. This means that you are born with it, and it's there. And so we have this. It's actually a huge area of research because it is tied to autoimmune disorders and just plain immune function. However, kind of what we think of later in life or later in the tree of life, adaptive immunity evolved. And so this is in vertebrates only, so us. And this is like your B and T memory cells. And this is a slower and specific response to pathogens. So this is only in specialized cells and tissues, and it does increase with repeated exposure. So these cells are quite literally changing their genomes to then respond and memorize pathogens. In vertebrates, adaptive and innate immunity kind of act in synergy. So usually the innate immunity signals to the adaptive that there is an invader. And then the adaptive cells can help facilitate immune function and then memorize this pathogen to protect from later infection. So there's this constant crosstalk between these different layers of immunity that have evolved in vertebrates. Regardless, innate immunity, or most organisms, like I already have reiterated, only have innate immunity, and they live for hundreds of years. They do fine. And so this is trees. Plants only have innate immunity. They have no sort of adaptive immune response. Lobsters, flies, invertebrates of many different kinds, they all have this specific innate immunity response, and they do not have. So, is adaptive immunity really required? Well, now it is because, but anyway, this is what vaccines take advantage of, is this adaptive immunity, because you can inject, certain molecules that then your cells can then recognize and help prevent later infections. Whereas innate immunity would never do that. And so all organisms in the tree of life have innate immunity. So what does this actually mean? This means that the same protein families that serve to recognize and respond to invader are found in most organisms. And the response is generally the same. And so here I'll just kind of spotlight a couple. So pattern recognition receptors recognize general pathogen markers on the cell surface. And this is a family of proteins that can be a little more diverse depending on what organisms you're looking at. But basically they function similarly, where they recognize certain patterns of pathogens, like the flagella. Like we don't have flagella, and this specific pattern recognition receptor recognizes flagella. This then starts an immune response where the cell will then upregulate maybe secondary metabolites or certain other things that can help protect itself from this pathogen. But the pathogen usually has, it wants to get in the cell, so it has some mechanisms, and this is what we call, actually, I forgot what it's called. Basically, they're combating, and they've been combating over time for millions of years. And so the microorganisms try to quell this response. They try to stop the immune response. And so they'll usually inject proteins into the cell to stop the immune response. However, there is another type of recognition, which is intracellular, called NLRs. And these are nucleotide-binding domain leucine-rich repeat proteins, which is a mouthful, so I'll just call them NLRs for the rest of the time. And they're intracellular receptors that are known to recognize infection and damage of a cell. And so these proteins are specific to recognizing, the invasion of the cell. And so I wanted to kind of just reiterate that this diagram that I'm showing right here is general enough to be an animal cell, even though it is a plant cell. And so here I'm showing the two different, well, basically the same protein, these NLRs, but in plant and animals. So you can see that they're highly conserved. They're basically the same protein, and they basically function in the same way. Most NLRs instigate cell death, and cell death is a common phenotype of immunity, which is kind of weird and convoluted. Why would you kill a cell? Why would you want to die if you're being invaded? It seems kind of backwards, right? But the fact is that these pathogens want to use your cell as niches of replication. They want to infect the cell. So if you kill your cell, these are multicellular, like we are multicellular organisms. If you kill like one cell, then that's fine, right? And basically you stop the pathogen from replicating. And so a very common phenotype we see in immunity is just kill the cell that's being invaded, stop the pathogen from going to other cells and hurting you. And so, yeah, here I'm kind of reiterating this, this kind of main theme of immunity is that, oh, shoot. And here we'll kind of wrap back up through the genetics we talked about. But if you cannot recognize and adequately respond to a pathogen, it can use your cells as niches of replication and take over. And so we just talked about NLRs. Here is showing NLRs that are a sect of different organisms that have the NLR versus a sect of organisms that don't have the NLR, meaning that these are genetic knockouts of this specific NLR, or a variant, a natural variant that doesn't have it. Anyway, doesn't matter. Basically, here we see that with the NLR, the plants are healthy. And basically what this translates to microscopically is that once the pathogen tries to infect the cell, sorry, I just lost my notes, but that's okay. I can just go off of memory. Once a pathogen infects a cell, the cell dies. And so then you don't get any replication. Whereas if you do not have the NLR, you see susceptibility of the plants. They're all just decimated. If you look microscopically, you see this specific pathogen was powdery mildew, which is a fungi. You see the hypha growing through the cells and excreting its hostorium, which is basically a cell used in powdery mildew to take nutrients from plant cells. And it's growing within the cell. So if it's not able to recognize a pathogen, this pathogen can then use the cell. And this is the same with animals. Here we see kind of on a more of a tissue level, we see splotches of bacterial infection when there's no NLR to recognize. And the cells look more or less so intact here in your epithelial lining of your gut. However, if you zoomed in, you could see a bunch of bacteria in your cells. So this person is sick. Like it looks like your cells are fine, but they're sick. There's bacteria replicating in them. However, as you see here, you kind of see sloughing of cells, cell death. And so this is just to kind of reiterate that one of the main phenotypes of immune response is cell death. And that helps limit the spread of your pathogen. And so fungi are known to be hosts of a variety of pathogens and parasites, such as viruses, bacteria, other mycoparasitic fungi. So even in their same kingdom, they have, you have predation of fungi on fungi, and grazing nematodes. So this is, very analogous to all the pathogens that infect plants, all the pathogens that infect humans are kind of, it's being elucidated that everything that infects other kingdoms are also infecting fungi. And so, yeah, this is just showing bacterial infection and nematode infection of fungi. And yeah. So regardless of pathogenicity, fungi coexist and interact with bacteria in many environments. So now I'm going to kind of focus on bacteria and fungi. You can focus on a ton of other interactions, but I just kind of wanted to, primarily focus. And so they are found, mostly most important for humans is the agricultural and forest environments. Well, it's also important for fungi. The lungs and digestive systems in us, we have, biofilms of fungi and bacteria acting together. We have bacteria and fungi in our oral cavity, and bacteria and fungi are really necessary for the productions of food, such as wine and cheese, tempeh, sourdough. These organisms are constantly interacting with each other in many, many different environments. And so I'm saying regardless of pathogenicity, because even if a bacteria is not hurting you, and the pathosystem is defined as this organism's trying to use your cells and steal your nutrients. Usually immune systems still interact with good microbes. So like our immune system interacts with our gut microbiota. And so this is just showing kind of the different modes of bacterial interaction with fungal hyphae. And there's a lot known. Basically, there can be destruction of fungal hyphae, just pure necrotrophy. Like these bacteria live on dead tissues, so they want to kill the cell first. However, there is bacteria that are more biotrophic that kind of need the cell to stay alive. And so they're constantly taking from the fungi and interacting with the fungi in a certain way. And this is, there's also a whole new field of bacteria that actually live within the fungal hyphae. So they kind of just grow and replicate with the fungi. They don't destroy the fungi in any sort of way. They might hurt the fungi, but basically this is also a very new and open field. And so kind of modes of pathogenicity in fungi are just being described. And so here I'm showing kind of the classic agaricus bisporus, which is your portobello mushroom, and kind of the different diseases that have been described in it. And this is because, this mushroom has been cultivated for so long that we can kind of describe exactly how it's being, eaten by other organisms or not. And this is showing that there's actually a range of what bacteria do for fungi. Of course, you have more disease symptoms that would then hinder the mushroom from producing as much spores as possible. But you also have a big thing with Agaricus bisporus is this specific Pseudomonas putata, which is this microbe that instigates mushroom primordial formation. So without this bacteria, you get basically way, way less mushroom formation. And so a lot of growers actually inoculate with this bacteria. It ingresses into the hyphae and it actually lives with the mushroom. And so it's a big deal. And the other thing is that all of these diseases you see here are all from the same genus of bacteria, Pseudomonas. So Pseudomonas, this is the same for plants. It infects and it helps, and it depends on the species. So, there's such a wide range of how bacteria interact with all organisms, and it can change just in the same genus. And so there are numerous studies that have defined fungal responses to the presence of other organisms. So we're trying to think about immune responses here, right? And so this is showing Fusarium, the fungi Fusarium versus Ralstonia, and both are tomato pathogens. So they have the same niche, is what we kind of think about. And here we have this knockout again. So basically this bacteria is producing this specific compound called rallomycin that the fungi recognizes. And you can see that the fungi has stopped growing near the bacteria that's producing this rallomycin. However, if you knock out this rallomycin, the fungi grows over the bacteria. It doesn't really recognize it. And then the other thing is that this Fusarium fungi produces this other molecule called bicaverdin, which you can see kind of accumulating here as this red metabolite. And basically this stops the bacteria from ingressing. So without recognizing the bacteria, the bacteria can then ingress into the hyphae, and the bicaverdin is not produced. So the main defense strategy of fungi so far seems to, induce morphological and chemical defense. Is cell death happening? Who knows? However, all of these studies, you can basically compare in an immune sort of thought to that the rallomycin that's being sensed by the fungi is sort of similar to the bacteria that's being sensed, right? And the production of bicaverdin is kind of similar to this immune part of this cell. So basically, what is going on in the middle? What molecules are sensing these things? And are there molecules that can sense the bacteria on fungi? All of this stuff is not really known. And so kind of the main part of my thesis, or the main motivation for my thesis, is that receptor signaling mechanisms underlying fungal responses to bacteria are understudied and poorly understood. And so this is right here, this is the fungi I work on in lab, Neurospora crassa. And this is it. It's conidia, so basically it's asexual propagule stage that has just grown one little hyphae. And you can see the bacteria within this as well. And so this is what I'm showing here, is this conidia becoming one little hyphae. And the reason why we use this, we can talk about it later, but basically it cuts down on cellular differentiation, which can convolute experiments and stuff. But anyway, and so basically, how is a fungi recognizing these bacteria? There's a bunch of diffusible molecules, and they're probably recognized in different ways. But it seems to be like once there is, attachment of the bacteria or interaction where the bacteria is physically, we definitely don't know that. We definitely don't really know this either with the diffusible molecules. It could be many, many things. Basically, like nothing is known how they interact. And so now that we know more about that, fungi have all these predators, and we can kind of then see, well, how we can kind of hypothesize, how are they, helping to protect themselves. And so in 2020 or 2016, actually, a study came out showing that nucleotide-binding domain leucine-rich repeat-like proteins, so these things that are critical, so I've showed that right here, critical in immune function in plants and animals, are diverse in the kingdom of fungi and quite abundant. And so this is a bioinformatics study, and this is showing different phyla and different classes of fungi. And this is the number of projects is showing the number of species in each class that was used, or phyla, or anyway, that was used to kind of investigate. And this column is showing the amount of fungi that had at least one NLR within their query sequence. And so, and so this little pie graph right here is showing the amount that was looked at versus the amount that had NLRs. And you can see that, more than half to more of that, of more fungi have NLRs quite, and a lot of them have, this study shows the amount of NLRs in each genome, and there are a lot. And so this is why sequencing the genome is important, because we can kind of start to figure out what's going on. And so again, plants and animals have these specific proteins, and there are some studies on these proteins. So 7 proteins within fungi that are NLRs have actually been through cell and molecular biology studies. However, this only goes through 3 different fungi. So Neurospora crassa, Podospora, and so it's just showing that there's so much to be discovered. But all of these NLRs function in cell death, programmed cell death, very similar to the immune function. But these are not to recognize anything like bacteria or viruses. What's known is that these help recognize when fungal cells fuse. If they're not the right partner, they'll go through cell death. And this is the way they go through cell death. And so basically there are 7 proteins out of thousands of NLRs that have known function. And so my main hypothesis is kind of quite simple for my thesis, is that fungal NLRs participate in an innate immune response like plant and animal NLRs. And so my thesis can kind of be separated into 2 distinct categories. What signaling molecules receptor molecules are being used to sense bacteria at a distance, and what signaling reception molecules are able to recognize bacteria in a contact-dependent manner, meaning the bacteria is taking over the fungi. And so now I'm going to go a little bit, into my own data, but I'm not going to go too into depth because it can get pretty hairy. But basically, focusing on contact-dependent, I'm using two different fungi and bacteria that are easily manipulatable and able to do genetics on, like knocking those genes out. There's lines of genetic knockouts already available. And so I chose this system, or that's the reason why I chose the system. And so Pseudomonas syringae DC3000 is able to colonize hypha of Neurospora crassa, which has been a fungi that's been used since the 1920s, and is as a sole nutrient source. And so basically what's happening here is that the bacteria is able to replicate on the hyphae alone. And so this is suggesting that there's extracellular biotrophy going on. They also saw that the hyphae didn't die when it came into contact with this bacteria. So there's definitely some sort of interaction going on where the bacteria is possibly keeping Neurospora alive to use its cells for replication. So it seems like a pretty solid pathosystem. And so Pseudomonas syringae is able to actively colonize Neurospora on LA, but not both minimal. And so this is just kind of reiterating the fact that all interactions are also environmentally dependent. And so, that's why people care so much about soil, because if the soil is crappy, then, plants are more susceptible to diseases. And so this is showing, I grew the bacteria right here for a little bit, and then I inoculated Neurospora, and I saw active colonization of the hyphae in this environment, whereas in this environment, I didn't. It's kind of still located in the spots where I put the bacteria. And this bacteria is glowing because there's a GFP protein within it, which is a fluorescent protein. And I did this synthetic hyphal assay where you put fiberglass on a plate, and this kind of is similar to fungal hyphae in the diameter. And so you can kind of see how bacteria swim in the different environments. And the bacteria is pretty not able to swim on Vogue's minimal. So this kind of alludes to the fact that maybe the bacteria or the environment is directly affecting the bacteria's ability. And it's not necessarily that Neurospora is stopping the bacteria from colonizing this environment. And so my future plans for contact-related signaling is I'm currently doing a bunch of phenotyping of Neurospora's response to bacteria in different environments, which includes growth inhibition, death, and chemotaxis. And so I've only seen it, the bacteria grows on the hyphae. That's about all I've seen. And so this is confocal microscopy, where the red is a dye that only uptakes into cells when the cell is dead. So when you see red dye, that means the cell is dead. And so here I'm showing kind of an interaction zone of the bacteria kind of fighting with the fungal hyphae. And if you look closely, you can see that some of the bacteria are also fluorescing red. So there's definitely some sort of like fight going on. And here we see certain hyphal compartments that are fluorescing red. So there's definitely cell death. And I'm doing a bunch of quantification of this. And so the goal is to do a time course RNA-seq on the fungi after bacteria. And then I can start doing genetics of elucidating exactly what's happening, kind of working backwards. And so, yeah, this is what this is. Now on to, almost done. Distant related signaling. So what is the bacteria and fungi doing from afar? And so exposing Neurospora to larger amounts of bacteria resulted in changes of growth kinetics, which is again environmentally dependent, macrokinetiation, and cell death. And so this is just like fancy words for phenotyping, but basically here with exposure to larger amounts of bacteria, Neurospora basically didn't grow like very well, and it stopped growing. And when I looked at it under the microscope, these cells were definitely dead. However, when this is the negative control here, where without the bacteria, the fungi grows fine, and I don't see cell death. And so there's definitely some interaction going on. In this environment, I saw again that Neurospora grew. You can kind of see it here, but you see kind of the vigor of this hyphae, how it's changed compared to the negative control without growing bacteria. And if you look at it kind of further, you see a bunch more orange. And these orange are the macroconidia, basically what I was showing here in this picture. Each one of these are little orange dots that are produced. Anyway, basically in this specific environment, this bogemonal media, I see upregulation of growth. And in this environment, I see downregulation of growth. And so growth inhibition of Neurospora on LA is dependent on the amount of bacteria. This is likely via diffusible molecules. And so here I have the bacteria and fungi in a co-culture, and I inoculated the bacteria for different amounts of days. So these graphs are showing the amount of days I let the bacteria replicate on the plate. And the longer I let the bacteria replicate on the plate, the more products it's kind of exuding into the plate. And so then I inoculated Neurospora and saw its growth. And so the blue graphs right here are showing that Neurospora, the longer the days are, the longer I inoculated or let the bacteria grow, the more growth reduction I get of Neurospora crassa. So it's definitely dependent on the amount that's within the interacting environment. I can then, before I inoculate Neurospora, to get the diffusible assay, it's quite a simple assay actually. You just cut the bacteria out of the plate. And I saw kind of the growth regain, but it still was quite detrimental to the fungus. For volatile, basically you have the fungus growing on a separate plate, and you have the bacteria growing on a separate plate, and you basically trap all the gases. So we know that, fungi can sense bacterial volatiles, and we know in some interactions that the volatiles inhibit the fungus. So I did this with this. It's basically a gas chamber. And I did see some inhibition of growth, but the conclusion from this is that the growth is definitely caused by more of a diffusible molecule and less of a volatile molecule that's in the air. So something that has gone through the matrix of the agar plate. And so a little bit more results is that adding diffusible exudates directly to the plate recapitulates the growth phenotype. And so I know it's definitely a diffusible molecule. And adding diffusible molecules specifically to these germlings, which are the conidia that I'm showing again here, killed them immediately. And so anyway, so my distance-related signaling future plans is to dissect if Neurospora cell death is a programmed cell death or not. Like, are these exudates just killing the cell because they're really toxic? Or is Neurospora sensing these and then able to kill itself to maybe, not contact the bacteria, so the bacteria can then manipulate it and all this, there's a bunch of hypotheses here. So basically we just need to start dissecting this. So is this basically I just said this is a chemical warfare, or is it a defense mechanism? And so this is showing percent survivability of germlings with exudate. And it's not looking good for the germlings. So basically this is just showing kind of what I said in the last slide, that these cells die. I'm kind of over time, sorry. So yeah, the way to kind of figure this out next is to do RNA-seq and a bunch of different phenotyping on a bunch of different versions of Neurospora, basically cultivars of Neurospora, see if I get sort of a difference in survivability. And so, yeah, thank you. I just kind of want to show everyone that's kind of backing this project and give a good, just to show you that science is not ever done alone. Science is always done in collaboration with other people. It's not just me that's like doing the science and getting results. I have help from an entire community. And so these are all the people. These 4 people are within my lab. These two people, this is Louise Glass. She's studying Neurospora for her entire career. And her postdoc, Alex, who is like the fungal genetics king. He does a bunch of knockouts in different genes. He's great. And Morgan is more of a bacterial fungal interaction expert. And so talking to her has been instrumental. She studies a specific bacteria that integrates into a hyphae of rhizopus, which is a, mucormycotina fungus. And so, I'm just to kind of reiterate and give my thanks all around to people. And yeah, if you have any questions, or if you have questions for later, my email is down here. I also have, anyway, thanks. That's it.
Thanks, Grace. That was excellent. There were a few questions that came up.
In the chat.
In the chat, yeah. And I wanted to see if, Baron, if you wanted to, because you had several of them. I wanted to see if you maybe wanted to ask them, unmute yourself and ask them in person. And they sort of came up at different points in the presentation, but they were great questions.
Oh, great. Endocellular biotrophy, another beautiful form of symbiosis? Question mark. Yeah, I would say that endocellular biotrophy could mean that it is a symbiosis. There's actually definitely studies out there showing that certain bacteria help the fungus grow. And then they use the nutrient source. So it's definitely a symbiosis. However, there are some bacteria that definitely take advantage of the cell, right? So that would be more considered a pathogen system. This teeters kind of a fine line between what's a pathogen and what's a symbiosis. And usually once they've co-evolved for a long time, a pathogen could easily turn into a symbiotic partner, maybe. Who knows evolution, but there's a lot of studies that suggest this. So yeah, cellular differentiation is good for a clean lab-based study, but do results scale up to a real world? Baron, would you like to explain your question if you don't mind? Cellular differentiation. Are you asking about how the cell responds to the environment?
I think I remember when they typed that, Grace, you had said that you try to limit differentiation experiments because it confounds the system.
Yeah, so this is a good point. Yes, it usually actually does scale up because in each cell you have the same genetic code. You definitely have to verify that the phenotype you're seeing is seen in all cell types if you want to actually scale it up. But usually you do see this. And that's because, the cell kind of uses the same mechanisms in each cell type for certain things. Obviously, this is not holds true for all biological functions, but specifically for immune systems, specifically these proteins, usually it translates. So yeah, no NLRs in Saccharomyces. Yep. The theory of this is that they're single cell. So if you kill your cell and you're not multicellular, yeah, what you gonna do? You're just dead. And so it's mostly seen in filamentous fungi that have multiple cells. Do you see the opposite where fungus attack? Yeah. So I just got data where I show that Neurospora is actually definitely limiting bacterial replication. It does not want. So there's definitely a fight back. I didn't show that because it's kind of new. I didn't show a lot of data. Oh, yeah, thanks. Thanks, guys. Oh, thank you. Okay. Oh, yeah, follow my Instagram.
Yeah, does everyone have Grace's Instagram? I can drop it in the chat.
Yeah, I'm teaching this semester, so I have been definitely not on Instagram, which is quite unfortunate, but hopefully next semester I'll have a lot more time for it. And so I would love to connect with people and answer questions. I always love talking about people's interests. You know, there's just so much to learn all the time. It's an infinite amount of knowledge out there. So, you know, you can learn from me, I can learn from you. It's a great time.
Yeah. I wanted to, this isn't specifically related to your thesis, but I wanted to see if you might share for people that are, you know, wanting to get into the field of this type of research. And I saw that you were a fellow through the Sonoma, or you received like a fellow or a scholarship through the Sonoma Mycological Society. And, you know, I think, you know, we want to start offering programs like that as well,
Right,
to help, you know, help advance research in different areas of mycology. And so I just wanted to see if you might share a little bit about, you know, how that worked and your path. Just go into depth a little bit more on your path.
Yeah, so specifically for mycology, sadly, Texas was definitely not a great place for it. Hopefully this will change. I feel like your specific organization has helped instigating this change. But I definitely knew that I would have to move out of state. And so basically, you know, fungal biology specifically is not just cell and molecular biology. It spans all fields. So, you know, ecology, biotechnology, sustainable food, you know, there's a bunch of environmental studies that are used with fungi, like bioremediation and stuff like that. And so knowing kind of what you're interested in fungal biology is how to start. I was always interested in understanding, you know, how organisms interact. And so this definitely kind of steered me into more of like a cell and molecular biology path. And so I'm not sure exactly, but basically just kind of discovering like what research is being done out there. And this is in a lot of, you know, academic settings, you can kind of go to a website and see, you know, different topics in fungal biology that different labs are studying. Specifically for me, you know, fungal biology is kind of a niche field, but it's expanding rapidly. It's definitely like, it's definitely, you know, kind of at the forefront right now. And so there's not a lot of labs, but I estimate in, you know, the next 20 years or so, there'll be, you know, a lot more avenues. And so just kind of first understanding what I was interested in on like a very general level, fungi and cell biology, and then kind of like finding an area where I could study that. So I applied to grad schools with this very specific need in mind. Sadly, actually, Berkeley mycology is dying, and there's not a lot of funding for ecology here anymore. But you know, at different colleges, there are, like, for sure. And I applied to ecology programs as well and looked kind of like what fungi are showing up at what point. And that's what my rotation project was on. But you know, that professor retired like after I did my rotation. Like he was not going to take me on as a grad student. And that's fine because I was always more interested in cell and molecular biology. But I'm you know, I'm just saying that kind of knowing where you're at and looking at there's a lot of avenues. So it's kind of difficult. But for me specifically, it's been primarily academic. So going through school and applying, and yeah, I'm still a student, right? Like I am still learning so much every day. And so I know and I am hopefully becoming more of an expert in the things that I really enjoy. But you know, it's just, yeah. This schooling is teaching. It's less about like going to class and learning things. and it's more independent. So you're learning things on your own. And, or it's not really, there's a lot of collaboration specifically for skills. Like my lab has a bunch of, molecular biology skills. And the glass lab has been doing fungal molecular biology for a long time. And so kind of learning from them is what grad school is all about. So now I become, kind of familiar with certain protocols that can help me down the line in my scientific career, kind of know what's out there. It's an infinite amount of skills and like knowledge that you can use. And so it's just, yeah, anyway.
Yeah. And do you see this emerging field of fungal mycology sort of being something that's part of like the sort of private sector, or like, there's a lot of like biotech companies and like these food companies that are probably interested in these sort of things, but are they equipped, or are they, do you see the future having positions for people like you to then apply this in that kind of setting?
Yeah, totally. A bunch of biotechnology is centered around cell biology. If you know how a cell works, you can then kind of manipulate it in the way you want it to. And so definitely I see kind of a place for me there. But also just, community science, it's not just academic, is really on the rise in mycology. And they're really, all these community scientists are really filling in knowledge gaps that an academic setting would not fund necessarily. Kind of like just observing species. That's no longer like funded very much anymore. So I feel like there's room for all different types of people from all different types of background to help fill in like fungi. So you don't have to go through an academic path. However, like it's really rigorous. Like you really get to learn a lot. So, academia exists for a reason. And, a lot of community science was built on academic studies. And so without, the discovery of the ITS region, a lot of these fungal barcoding projects could not have occurred. There might have, and so like, and these are published works. And so academia is all about, publishing your work and making sure that it's accessible to everyone. And so, yeah, I don't know. There's definitely room for everything.
Yeah, so Emma.
I have a lot more of, yeah, there seems like amateur scientists in mycology. Yeah, I wouldn't call these people amateur scientists. They're definitely real scientists. But yeah, it's definitely still in its infancy. And I feel like specifically sequencing is like a big thing for, the citizen scientists. And I feel like once we have more tools, we can start even sequencing full genomes. It's like we're not just going to be sequencing like one, nanopore sequencing is an example. It's quite cheap. Maybe even, other generations of sequencing might become cheap in the future. Because, like the sequencing that they're using now used to be so expensive. Like it would have never been accessible to, but as we see technology progress, like there's just going to be niches for everyone to like get involved.
Yeah.
So, yeah.
We definitely want to do a class on either have invite Alan
totally
to teach us like, some of the things that are advancing with like the mini PCR and some of the newer tools that are accessible to citizen scientists and amateurs.
Totally. Yeah. And it's a great way to really understand like the basics of biology too, is to really get your hands on these organisms and see what's in their DNA. And yeah, it's pretty cool.
Yeah. Did I miss any questions? Is anybody out there that wants to pipe in that didn't get their question answered?
I have one question. There's a couple in the chat that you can take before mine if you want.
Oh, no, go ahead.
Grace, with the RNA-seq you're hoping to do, is that on the fungi or the bacteria too?
It's on the fungi. The bacteria is being done by someone else. Specifically what I'm doing is the fungi, but yeah. both sides would be important, but because I'm just looking at signaling molecules, and because I only have like 2.5 years left of my PhD, you kind of have to focus, you know? Yeah, so yeah, just the fungi.
Cool. Thank you. Great
Yeah.
talk.
Oh, thanks.
Yeah, very informative. Anybody else that we missed?
There's a lot. Do any of your peers in fungal mycology work in soil or water remediation? Jack works in remediation, actually. Yeah, that's Jack. There's a lot of academic people that work in how fungi break down like difficult products. Maybe not so on a large scale, but you know, kind of understanding entomology of what fungi excrete and what they break down is definitely a big academic like spot of interest. Oh yeah, Jack just put his email if you'd like to talk to him. He's definitely more of the expert.
Yeah, Jack, where are you? If you don't mind if I ask.
I live in Olympia, Washington.
Okay. Oh, great. And did you do this path through academia, or was this self-taught, or
I don't want to hijack Grace's talk, but
we can be quick.
Oh yeah, it's important.
I studied aspergillus, breaking down biodegradable plastics, and then I did a program with the National Science Foundation interviewing bioremediation companies, and I've just connected with lots of folks who have sat around growing fungi in buckets with oils and dyes and all sorts of things. So
Nice. We'll definitely take down your email. It's a topic there's a lot of interest, as Grace mentioned.
would love to connect.
Yeah. We've done one sort of talk on bioremediation using, studying the breakdown, using fungi to break down like invasive species here.
Oh, like plants?
Yeah, so like ligustrum. So
oh, cool. Yeah, I hate that thing.
Yeah, it's just everywhere, and
Yeah, it's
really, you can
everywhere.
really see it this time of year because it's the only like green in the forest, or not, the junipers and the
Yeah.
ligustrum.
It's all of the berries everywhere.
Yeah, but yeah, Jim Stack, who's a mycologist now in the Colorado area, was able to do a study, and it broke down the and return the nutrients back to the forest 11 times faster than just sort of sitting and waiting for bacteria and all the other sort of things that slowly break things down in the forest. So he used turkey tail inoculations. But yeah, there's so many applications there, and a lot to learn. So yeah, let's see. Anyone else have questions?
Yeah, it's really nice seeing all the Pacific Northwest people show up. Yeah, I think Emma put that this information that the Bay Area Applied Mycology is doing a similar project with eucalyptus.
Oh, nice.
Remediation, yeah. Just wanted to highlight that.
Yeah. Just kind of scanning through.
Oh, okay. Sorry, I'm definitely missing questions. Okay, so Emily had a question. Question from a non-biologist. What will we better understand in other applications from better understanding fungal immune responses? What a good. So this project is funded by the NIH, and it's like of high interest. And this is because these proteins are also found within us. And the issue with studying innate immune systems within us is that it's convoluted by the adaptive immune system. And so a big reason why people want to study like what these proteins are doing in fungi is because then it's very, directly related to what they're probably doing in us. And specifically, people have been looking at plants for a long time because plants are an easy pathosystem to define. Like you can see a sick plant pretty easily. Sick mushroom is kind of like, you know, who knows what's really going on. And so people have been using plants to study innate immunity function in humans for a long time. But fungi are more closely related to us. They're, you know, on average share 30%, we share 30% of our genomes. And plants are much, much more distantly related. And so, and, you know, fungi are more easily manipulated in culture and, you know, all this stuff. So yeah, that's the main application. Why we get funding, of course, is like, has to be applied to humans. But basically, just understanding how fungi biology functions is another huge thing. Like it's a rule of life sort of thing. Like, how does life function, period? And that's like of high interest, because then you can kind of understand how fungi might be interacting with other organisms. And that's like an important thing to understand. And so, yeah, it's just basic biology stuff. Yeah.
Appreciate you explaining to a non-biologist.
Yeah, did that make sense? I hope that makes sense.
100%. Yeah, thanks.
Oh, great. Yeah.
Yeah, there seems to be such a, there's all these tests that you can start to take to like try to understand your microbiota, but then like the interpretation and then sort of like changing the inputs or like doing something about it. Like there seems to be like kind of holes there still.
Oh yeah.
But like this type of research seems, I was thinking about that a lot. Like, so I've done a few of those tests and it's like, oh, that's cool. I've got all these yeast and bacteria, but,
Yeah.
but how are they interacting? Like,
Right.
it's like, it's.
And for a long time, I mean, this started only about 10 years ago, is this microbiota research, right? Everything had been studied kind of azenically, meaning it was one organism in the plate functioning, and that has been biology for hundreds of years. In reality, organisms never live azenically. Like they're always interacting with other organisms, including us, right? And so biology obviously changes like so much when you start interacting with everything else. So yeah, it's a new field, this microbiota field, and putting one fungi with one bacteria is already really complex, right? But in reality, it's like 4 fungi with 18 bacteria.
Yeah.
So, and they're interacting with each other, and yeah, it's just it gets pretty convoluted.
Yeah, how complex can you get sort of in a lab setting when kind of showing these interactions?
So the kind of research that's being done now in big, people care about microbes in the context of agriculture. And so there are actually studies where people will grow plants and inoculate the roots with like a whole entire community of bacteria, and they kind of see what bacteria drops out, what bacteria stays, and these kind of community studies. However, there's not a lot of studies where they put bacteria and fungi together, because then it just gets like so uninterpretable that they don't really know what's going on. It's obvious, like if the plant grows better, then maybe that fungi and that bacteria help. But how is that happening? Now you have 3 different interacting partners. You have bacteria interacting with bacteria, you have bacteria interacting with fungi, you have bacteria interacting with the plant, you have plant interacting with fungi. It's just like, that's really how biology has always worked. But that stuff is just not really touchable by the science tools that we have right now, and kind of understanding how it functions. But people do like make microbes to put onto a field of agriculture, knowing that these microbes will help. And that's important, because that's like directly applied. Like you're going to save the field crop by putting these microbes out there. The end. Like we don't really care what the cell and molecular biology is and how they're doing that. We do care, but because it's really convoluted, maybe we'll care later on when we have better tools to kind of dissect. that.
Yeah.
But
And do you think sort of like sequencing the whole genome too will allow for, like, I'm sure there's like a lot that can happen with like machine learning. And I
yeah,
don't know like how I used to be in the tech field and kind of follow some of the types of projects.
right.
I think back then it was like big data. Everything was
Yeah.
like big data. But
My lab is actually a genomics lab because studying evolution of innate systems is like using, genomes and seeing what's there and what's not. But it's all of the machine learning and all of the biology that we know are based on cell and molecular biology studies, right? Like we know that this protein functions in this way because someone was in a lab, looking at this protein under electron microscopy, like defining like what it was. And now we know that the gene that prescribes this protein, probably does what it does, right? And we can kind of see that in different genomes, but yeah, big data is difficult to validate without actual cell work. Yeah, so we're kind of at a point where we can, predict a bunch of things with genomics, and specifically AlphaFold, which is a machine learning protein predictor. We can then use AlphaFold to predict what the protein might look like. But in reality, that's about as far as we can get. Is we can predict what it's doing, and then maybe we can you have to go into the lab and like see if it's doing what you think it's doing, right?
It can kind of get you in the ballpark maybe and help
Get you in the
come up with
ballpark, yeah.
hypothesis, and I guess like indicate or sort of like direct research maybe too.
Yeah. Oh no, totally, for sure. Yeah. I mean, a lot of my project is based upon people seeing that NLRs are within a lot of fungi. And that was a genomic study, like classic genomics, like, oh, here are all these proteins. What are they doing? Who knows?
Yeah.
But they're there. So,
Yeah. Well, very good. Anyone else out there have anything to add? It looks like Rachel asked, what programs do you use most?
Oh, you mean for bioinformatics? I am learning Python. But R is another one that I have used to make graphs. But I'm mostly trying to focus on Python right now, if that's what you're asking, right? I think so. But yeah, I'm more of a lab scientist, and so I am very new with big data genomic stuff. I'm excited to learn more though. What kind of interactions are there between lab research and applied field research? A lot, yeah. So I'm guessing you mean applied field research as you're going out into the field, kind of seeing what organisms are there, maybe what they're doing. I guess in the context of agriculture, what organisms are in the agricultural field. There's a lot, yeah. RStudio, yeah. I guess interactions. Sorry, I don't really understand your question if you wanted to ask it.
Okay.
I guess you're seeing is it, yeah, there's a lot. In order to kind of define phenotypes you might see in a field, it really does come down to like the biology of the organism usually, and how the biology is, how they're interacting with other things.
I guess I was just kind of wondering because it's sounding like I was thinking about how like lab research is, how you were talking about how you have like you're studying like the interactions between like two organisms, and it's vastly simplified compared to like the complexity of natural systems that are there in like the soil. And so when I'm guessing that there's also research on like a different level, like in the field, like in agricultural fields, when people are like trying out, to see if like inoculating with one specific thing like improves certain outcomes. agricultural outcomes, and but in the field they're dealing with these really complex systems, and so I guess it seems like it's a different kind of scientific study. I'm not a scientist. I don't really understand that much about how these things work, but it seems like one would inform the other and vice versa, and that there would be some sort of like feedback between the two types of scientific studies. I don't know what I was trying to ask you.
No, it's true. Yeah. I think I already answered it, but yeah.
Right.
Yeah, they do definitely kind of translate into one another for sure. They definitely inform one another. So looking at the cell and molecular biology of an organism without knowing that these organisms like were in the same niche, kind of, you might not get any results, and vice versa. Even though it's complex, like the microbial interaction, like in the field, if when you start stripping it down and you start, interacting with them pairwise, and you kind of see what is going on between each organism, you can kind of start to dissect it. And specifically with like cell biology, you can really kind of understand what's at the basic level of how these organisms might be interacting. So they definitely inform each other. Yeah.
Nice. Well, if we don't have any other questions, we'll sign off here in a bit. And we thank you so much for gracing us with your questions. It's like, I'm sure you hear that all the time.
Yep.
I have a name that gets turned into a pun a lot too.
Angel, yeah. Thanks.
Yeah, next time you're in the Central Texas area, we'll have to go look for orchids or mycoheterotrophs.
Yeah, definitely. I'll be there in March. I don't know if anything.
I know the spring floral roots come up in February, March, maybe. Just depends on what kind of year we have.
Right. Okay. Cool. I'll definitely always down. Yeah. Thank y'all. Yeah. If you have any questions, please email me. Or if you want my PowerPoint slides for whatever reason. Yeah.
Great. Well, thanks again. And thanks everybody for tuning in. And as I mentioned, our December program is on the fly agaric. And the event link is up on our website. So we also have another in-person workshop coming up too on December 9th. So it seems like everything falls on Thursdays through the month of December. So keep your Thursdays free to hang out with the Mycology Society. But yeah. All right, everybody. Thanks for tuning in. Thanks, Grace.
Yeah. Have a good night, everyone. Okay, bye.
Bye.